Firstly, I'd like to thank the organizers for the opportunity to participate in this symposium. I'm excited to be able to talk to you all today about the concept of the food matrix. In this presentation, I'll be highlighting the complexity of food and encourage you to look beyond the nutrition facts label and consider the food matrix. This slide lists my disclosures for the past 12 months, including my employment at the University of Illinois, my grants, and speaking honoraria. I'd like to begin with a brief outline of what I'll be discussing in this presentation. In the first part, I'll introduce the concept of the food matrix and provide examples of food matrices' impact on nutrient bioaccessibility and health outcomes. In the last part of the presentation, I'll provide a few examples of matrix-specific health effects and plausible biological mechanisms of the health effects. As nutritional scientists, we all acknowledge that diet affects health, including cardiometabolic health, adiposity, and neurocognition. Historically, research has focused on reductionist, nutrient-driven approaches to understanding the effects of diet on health. However, increasingly, research is indicating that the consumption of whole foods, as compared to isolated nutrients contained within foods, can have surprising effects, and that the whole is often greater than the sum of its parts. Ultimately, we eat foods, which contain nutrients. Therefore, further exploration of these phenomena are warranted so that we can better understand the nutrition-health interface. So let's consider the concept of the food matrix. The food matrix is defined by the USDA as nutrient and non-nutrient components of foods and their molecular relations. The food matrix is thought to contribute to diet-health relationships, and my group is particularly interested in this as complex food matrices such as those within whole grains or nuts, can reduce the bioaccessibility or digestion and absorption of nutrients, thereby contributing to the substrate availability for the intestinal microbiota. As the microbiota can metabolize non-digested food components often found in complex matrices, like fiber, polyphenols, and/or bioactives, it has the capacity to significantly influence health physiology, therefore helping connect diet to host health. Let's consider the complexity of foods a bit more, noting that both food science and nutritional aspects of the diet can affect health outcomes. Ultimately, foods contain nutrients that vary in their bioaccessibility. And by bioaccessibility, I'm referring to the fraction of an ingested compound, be it a nutrient or bioactive, which is liberated from the food matrix within the gastrointestinal tract. This figure highlights some important components of plant matrices, including the different types of fibers within the plant cell wall and the encapsulation of starch granules within plant-based cell matrices. As we know, fibers are resistant to digestion. Thus, plant-based nutrients will vary in their bioaccessibility depending on external factors, such as milling or cooking, to disrupt the plant cell walls to liberate nutrients, like starch, for digestion and absorption. However, some digestible polysaccharides may be inaccessible to digestive enzymes within the food matrix due to the cooking process. For example, resistance can be conferred following cooking and cooling, resulting in retrogradation of starch molecules. This can affect physiological responses, such as the glycemic response to the ingestion of pasta that's been cooked and then cooled. Ultimately, the different molecular configurations in dietary fibers results in their different physicochemical characteristics. External factors like temperature, pH, mechanical disruption, and fermentation also affect the food matrix. As we just discussed, heating and cooling can make starch resistant to digestion. Acidity can denature proteins in meat to facilitate the digestion and absorption of amino acids. Grinding almonds into a paste disrupts the plant cell wall, increasing the metabolizable energy content compared to whole almonds. And utilizing microorganisms to ferment milk to produce yogurt changes the physical structure from a liquid into a gel. Ultimately, food matrices affect digestion and absorption. The physical structure created by a combination of nutritive components within the food can act independently of its individual components during digestion and absorption. Compared with an isolated nutrient, certain matrices protect their nutrients and bioactive compounds against degradation. Food matrices affect digestion and absorption, and this degradation, or lack thereof, will also affect substrate availability for the microbiota within the intestinal lumen. To summarize this portion of the presentation, the food matrix, or the physical domain that contains food components, like nutrients, provides functions that are different than if the nutrients were consumed in isolation. Matrices affect the bioaccessibility of nutrients, meaning that the food's ability to be digested, absorbed, and fermented is impacted by the physical structure of the food, as well as the external factors that have acted upon it prior to consumption. Increasingly, in vitro, preclinical, and clinical research is demonstrating that the food matrix effect has physiological implications, such as the metabolizable energy of a food and the effects of consumption on health outcomes, such as blood glucose and cholesterol concentrations. It's important to look beyond the nutrition facts label and consider the food matrix, because the nutritional content of foods, be it eggs, grapes, nuts, or milk, will vary based upon how the original matrix is modified through cooking, grinding, or fermentation. Now, I'd like to walk you through a couple of case studies on the relevance of the food matrix. I'll begin by first discussing the effects of processing on nutrient bioaccessibility of almonds. Nuts are nutrient-dense foods that contain fiber, unsaturated fatty acids, and polyphenols, along with other types of fats, protein, vitamins, and minerals. Epidemiological and interventional trials have demonstrated that consumption of nuts contributes to benefits to cardiovascular health, glycemic control, and body composition. To better understand the effects of processing on the metabolizable energy of almonds, which may underlie some of the associated health benefits of nut consumption reported in the literature, my colleagues at the USDA Agricultural Research Service in Beltsville, Maryland, designed an elegant, complete feeding crossover design trial to determine how roasting, chopping, and grinding almonds affected nutrient bioaccessibility. Adults participating in this study underwent 5 study conditions, which differed only by the inclusion of different forms of almonds. After each 3-week condition, the participants underwent a washout period prior to randomization to each subsequent condition. Let's first look at what they reported with regard to how processing affected the physical properties of the almonds. Figure 1 from this publication is images from the fracture of 1 whole natural almond, 1 whole roasted almond, or 1 gram chopped roasted almonds after compression. These experiments revealed that the whole natural almonds had a greater hardness, but a lower number of particles per image compared to whole roasted almonds. So the physical properties related to the processing of the almonds ultimately affected the metabolizable energy of the almonds. This table shows the almond type, its measured metabolizable energy, and its estimated metabolizable energy using Atwater factors. What you should note is that the whole natural, whole roasted, and chopped roasted almonds provided less metabolizable energy than predicted. This is important because it means that these undigested nutrients were available for microbial metabolism. And this is where my research laboratory was able to come in and help to expand our understanding of these results by characterizing how almond processing also affected the intestinal microbiota. This figure shows that the consumption of roasted chopped almonds increased roseburia. There was also a numeric increase within the whole roasted condition and the whole natural condition. Notably, almond butter was almost identical to the control. This is interesting because it suggests that disrupting the almond plant cell wall through roasting and grinding into butter liberated the nutrients in a way that makes them accessible to human digestion and absorption, thereby limiting the availability of nutrients for the microbiota. Other bacteria, such as Lactospira and Dialister, had similar response profiles. In summary, almond processing impacted the nutrient bioaccessibility. Almonds had up to 25% less metabolizable energy than predicted by Atwater factors, and consumption of different forms of almonds differentially affected the human fecal microbiota. Let's move on to a different case study and consider the relevance of the dairy matrix on health. Firstly, dairy is considered to be an excellent source of high-quality protein and calcium, as well as other vitamins and minerals. There are three main types of dairy matrices: liquid, semi-solid or gel, and solid. The dairy matrix also contains different bioactives, or food constituents other than those that meet basic nutritional needs that are responsible for a change in human health. Examples of bioactives in dairy include the milk-fat globular membrane, which contains the different fatty acids, and bioactive peptides, which are protein fragments produced via modifications or cleavage from parent proteins. Let's first look at an example of how the bioaccessibility of fat is affected by the dairy matrix. This figure on the right shows the significant increase in fecal fat excretion in 11 adults that participated in a randomized crossover study that compared two diets, one high in calcium from low-fat dairy products and one low in calcium. These results indicate that increasing the intake of calcium from low-fat dairy products doubled the total fecal fat excretion. The results may partially explain why a high calcium diet can help contribute to weight loss. The high phosphate content, as well as the presence of the milk-fat globular membrane, are also hypothesized to contribute to health effects of dairy consumption. These health effects include associations reported in epidemiological studies and results of intervention trials demonstrating that yogurt consumers, for example, have lower body weight than non-consumers. And changes in blood lipids is thought to be a reflection of the effect of calcium on bile acid reabsorption, which leads to the de novo synthesis of bile acids from plasma cholesterol as a substrate. Moving on to focus more specifically on yogurt. Yogurt is created through microbial fermentation of milk by the lactic acid bacteria Streptococcus thermophilus and Lactobacillus bulgaricus. Fermentation results in the gel-like structure of yogurt and contributes to the presence of bioactives, which have associated health benefits. For example, some dairy-derived tripeptides have been shown to have increased angiotensin-converting enzyme inhibitory activity, thereby contributing to reduced blood pressure. Exopolysaccharides, produced by lactic acid bacteria, play a structural role in the yogurt matrix by increasing its viscosity. They may also contribute to gastrointestinal immunity. Beta-galactosidases, derived from the live microorganisms in yogurt, contribute to lactose digestion within the intestines, providing a notable benefit to lactose maldigesters. I'd also like to briefly discuss the relevance of the matrix for probiotics. Probiotics are frequently added to fermented dairy products like kefir and yogurt. Some of the strain-specific health benefits as reduced duration of diarrhea and improving constipation. What I find particularly interesting is that there's an interaction between the dairy matrix and probiotics, in that in vitro research has demonstrated that probiotic survival is better within yogurt as opposed to free probiotic cells or probiotic cells that have been microencapsulated. Some of the proposed mechanisms for this improvement in probiotic survival include the contributions of the yogurt to buffering stomach acidity, the presence of lactic acid bacteria-derived exopolysaccharides and milk-fat globular membranes offering protection in the gastrointestinal tract, or the presence of lactose as an energy source for the probiotics. Let's take a moment to summarize the concepts related to the dairy matrix. Firstly, we discussed the contributions of calcium within dairy foods on increasing fat excretion, which may underlie the matrix-associated health benefits as they relate to cholesterol concentrations and body weight. Specific to probiotic survival, the provision of probiotics within yogurt has been shown in vitro to enhance probiotic survivability, which may have relevance to the demonstrated health effects of certain probiotic strains that are added into yogurts and other fermented dairy products. Other bioactives present in the dairy matrix include the presence of beta-galactosidases from live cultures in fermented dairy, which contribute to lactose digestion, tripeptides having antihypertensive effects, and exopolysaccharides contributing to functional and health properties of yogurt consumption. In summary, I hope that this presentation has provided you with some insights as to why it's important to look beyond the nutrition facts label and consider the importance of the food matrix as you conduct and evaluate science related to nutrition and health. Food is complex, and the physical structure of the food will affect nutrient bioaccessibility and ultimately human health. It's an exciting time to be a nutrition scientist because of our growing understanding of the food matrix and matrix health connections. Thank you for your time and attention, and I look forward to further discussion at the end of the session.